So work package 4 focuses on the impact of endocrine disrupting chemicals, so EDCs, on isolated organs as well as physiological barriers. In order to do so, we are using in vitro advanced technologies, for example, barrier systems as well as zebrafish and organoids. And this helps us to understand the impact of EDCs on the barrier integrity as well as on the toxicity in specific tissues, and then also the impact on specific organs and therefore the impact on diseases. My focus in the project is on placenta barrier. Placenta barrier is important for the difference between the maternal and the fetal circulation. So we use two different cell types, endothelial cells as well as trophoblast cells, and they are seated on a transwell in order to mimic the human placenta barrier. And then later on, we also want to introduce more advanced models, for example, organoids and spheroids of placenta, to see the bigger picture. And in addition, other partners of Work Package 4 are looking at several other organs, as for example, the blood-brain barrier or the liver and the intestinal tract. And therefore, we have a look not only at the barriers, but also at the metabolism, the uptake, and the distribution in the human body. In our model, we use staining in order to see whether the viability is changed with the impact of endocrine disruptors. So, for example, we do a dead live staining in immunofluorescence. Then we are also measuring the TEER value. TEER stands for trans-endothelial electrical resistance. And here we have two electrodes which go on the maternal and fetal side. And then we measure the resistance over the barrier. And if the value drops, the resistance drops, and therefore we can assume that the barrier is a bit more leaky than before. To measure the transport, we are using fluorescence-traced molecules, for example, phyzidextran as well as sodium fluorescein in different sizes. So we put these molecules on the maternal side, the apical side, and then measure the fluorescence in the supernatant on the basolateral side. And therefore, we get an idea if these different sized molecules can pass through the barrier over time. Furthermore, we do staining for the tight junctions. So here we can see whether the adherence between the cells, so the integrity over the barrier, is changed. Our Work Package 4 is part of a big puzzle of ENDOMIX. So we are collaborating with all the different partners of the other work packages. For example, Work Package 2 does identify the chemical mixtures and therefore uses the cohort studies of Work Package 1. And then Work Package 3, which has a look at the immune cells, they are using this mixture in order to treat the immune cells. And then in our case, we are taking the mixture from Work Package 2 as well as the immune cells from Work Package 3 and add them to our barrier systems and organoid models to further challenge them. So all in all, our Work Package 4 acts like a bridge between the cell-based studies and the in vivo studies, looking more into the organs and the barrier systems. And then further on, Work Package 5 uses our outcome to check the cohorts again, and Work Package 6 looks at the correlation between in vitro and in vivo. So all in all, Work Package 4 brings together the different pieces looking at the organs and the barriers.